Sharing the screen. Okay, can you see that properly?
Yes.
I'm assuming yes. Okay. Yeah, good morning, everyone. And yeah, thanks for taking the time out to listen to what we have to say about the technologies that we're using for our research at the moment. My name's Anthony Bernard. I manage the Marine Remote Imagery platform at the South African Institute for Aquatic Biodiversity, so SIAB. Essentially, what MARIP aims to do is develop and provide imagery-based sampling equipment, as well as technical and scientific support and data management infrastructure to enable research on seafloor ecosystems and marine animals to support the sustainable management of these ecosystems and species. We use a variety of imaging systems, and essentially what we mean by imaging systems is that they're underwater cameras of some or other sort, and this includes remotely operated vehicles, scuba-based diver-operated camera systems, baited camera systems, drop cameras, to name but a few. The main equipment that we use, however, are the baited remote underwater stereo video systems, so stereo BRAVs, the remotely operated vehicle, ROVs, and then the deep sea landers, which are a platform that gets the cameras to and from the seafloor. And I'll go into a little bit more detail about all of those over the course of this talk. One of the main things that we do within the platform is also work towards growing the field of research in South Africa and regionally, and we support research organizations, most research organizations in South Africa, and have a large cohort of students working through the platform and working on the projects. So there's a large component of capacity development as well as the technology development. So the field of imagery research in the marine environment has been around for about 70 years. It started in the mid 1950s, early 1960s, where underwater video was first used to survey or to survey underwater ecosystems and fish. This like pioneering type of work was a bit sparse in the beginning. There wasn't much outputs from this. And by the mid-1990s, with the advent of more reliable and safer scuba-based systems, most of the underwater research started being done with, or observation-based research started being done with scuba-based visual surveys. However, come the 2000s and the mid 2010, the technology development with digital cameras and improved robotics and remote operated vehicles meant that the video-based sampling equipment started growing in popularity, and more and more research outputs have been emerging from this field. So this figure basically just tracks the use of the different methods through time from the early 50s, 60s, where you can see a variety of different camera systems being used. But with the advent of that scuba-based sampling equipment, the underwater visual sensors, the green line, started dominating the field of research. However, recently, with the technological advances, we've started to see the baited camera systems, so BRAVs, and the ROVs, ROVs, remote operated vehicles, taking more prevalence in the scientific field. Okay, so what is a ROV, a remote operated vehicle? It's basically a radio or a vehicle you can fly underwater. It's got a cable that's tethered to a vessel, and then there's a pilot on the boat who can drive the ROV around. This enables exploration type research. It allows the researchers and scientists to collect specimens off the seafloor. That's then useful for taxonomy and species descriptions, but as well as bioprospecting. It allows exploration and exploring new habitats that were previously inaccessible. For example, the coelacanth work that South Africa has been doing in Sodwana Bay and KwaZulu-Natal. But then it also allows assessments of fish populations and invertebrate populations. So you can swim transects and collect quantitative data on the abundance and size of fishes. And then you can collect habitat data from areas and depths that are previously inaccessible. One of the main strengths of these tools, the ROV, is that you can use it across broad depth ranges, so from the shallow subtidal down into the deep sea, and this makes it much more applicable or much more flexible than the diver operated system. diver operated sampling techniques that are typically more shallow. The other tool that we use are the stereo bruvs, so baited remote underwater stereo video systems. And these are a relatively straightforward piece of equipment. You have camera housings that hold digital cameras of some sorts, typically GoPros, and these face onto a baited camera system, onto a baited container that draws the fish into the field of view. The bruvs are tethered to the surface, so they've got a rope that goes to the surface, and you can lower and deploy them off boats without having to get in the water. The neat thing about the stereo bruvs is that because you've got the two cameras, you can calibrate the field of view where the two cameras overlap. And using photogrammetry, you can then get the size of the fish that you see. This then becomes very useful for quantitative surveys of fish populations. You can look at the size frequency distributions, abundance, and general geographic distributions of the species. Because we're using bait, the method is obviously really good for surveying predators. So it's widely used for shark research and shark surveys, but it's also widely used to survey marine protected areas. It's a pretty straightforward and low cost equipment to operate, and it causes very little, it causes essentially no damage or harm to the marine environment. So there's no impact of the research. And then deep sea landers are the other piece of equipment that we have within our group and that we use quite widely. So a lander is essentially a platform that allows you to deploy scientific equipment or scientific payloads remotely to the seafloor. So without having to physically put the equipment there or without the equipment being connected to a rope. And the way it works is it just works on buoyancy. So when you deploy it, it's negatively buoyant. You have a ballast weight attached. And then it lands on the seafloor and it can stay down for essentially as long as you want. So from hours to months, doing whatever the scientific task it was programmed or designed to do. When it's time to retrieve the equipment, you send an acoustic command to an acoustic release and it drops the ballast, and then the system becomes positively buoyant and it floats back to the surface. So it's a very flexible and dynamic piece of equipment and allows you to collect an array of information over varying time periods. We've been primarily using it for stereo video systems, so we've designed it to be essentially equivalent to the stereo bruvs that we use in more shallow systems. And this allows us to get comparable data from intertidal zones down to like the shelf edge where we use stereo bruvs and down into the deep sea. So from 200 meters down to 3,500 meters. And again, this equipment is really useful for doing quantitative surveys of deep sea animals. So it's the fish, but then also the macroinvertebrates within the deep sea environment. A lot of the animals are scavengers, and they're actively attracted to food falls. So a baited camera in the deep sea is essentially an artificial food fall, and then you can see what animals are there, and then also monitor their behavior around that. And then this all starts to feed into marine protected area monitoring, species distribution mapping, and marine spatial planning. In terms of the research scope of MARIP, the ROV can operate from about 10 meters down to about 1,000 meters. We have two ROVs within the facility, and that lets us work up to about 25% of South Africa's exclusive economic zone. The exclusive economic zone is the marine territory that's within South Africa's management around our coastline. With the landers and the stereo bruvs, we can work down from the intertidal down to about 3,500 meters, and this gives us 61% research coverage of our marine territory. So there's still a portion that is missing in terms of our capabilities. However, we have made significant advances in being able to do surveys and collect quantitative data from these deeper water ecosystems. The platform has enabled us to do comprehensive surveys within South Africa, inside and outside of most of South Africa's marine parks. But then we've also made the platform available to researchers from neighboring countries, and we've supported research in Angola, Namibia, Mozambique, Madagascar, Kenya, Tanzania, Seychelles, Comores, and Maldives. So the platform's been widely used regionally, just showing the value of it. The platform, as well as so it's the equipment, as well as the technical expertise and scientific expertise that we offer. and we've been giving training courses to most of the people that we've been working with. In terms of some of the impact and the management support that the data that we collect can provide. So this is some results from the Beta Camera, the BRUV surveys, looking at the effectiveness of marine parks for protecting and supporting the management of important fisheries species that are, so in this figure, the green blocks show that there was a significant positive MPA effect. Red block shows that there was a negative MPA effect, and then grey, there's no effect. So with species like the Dacharat, you show that the MPAs are really important. For example, you see more of the species inside the MPAs. When you see them, there's a very high proportion of adults, and then they're more abundant and bigger. For some other species like the Englishman and Slinger and Santa, you see more outside the MPAs. However, when you come into the MPA, you see more adults, and then they either are more abundant or they are bigger. And this kind of demonstrates the value of marine protected areas in providing a refuge for individuals and species to grow to adulthood, where they can become, where they can contribute as spawners and contribute to the population growth and the maintenance of fisheries populations outside, or fish populations outside the marine parks. In a similar vein, this plot kind of shows the biomass of mature fish inside and outside of marine parks in KwaZulu-Natal. So on the left-hand side, you've got the Ponderland MPA, and then on the right-hand side, you've got the Isimangaliso MPA, and in between, you've got the fished areas and the newly proclaimed Tugela Banks MPA. And it quite nicely shows that the value of MPAs are protecting these fisheries targets. So you have significantly higher biomass of them inside the established MPAs, really low biomass of the targets outside the MPAs, where they are commercially exploited and exploited by recreational fishermen. And then it also shows that new MPAs take a while potentially to start to show benefits from protection. So an MPA isn't an immediate benefit, it takes time. I think that the Ponderland MPA is about 20 years old now, and the Isimangaliso MPA was originally proclaimed in the 70s, I think. So it's 30, 40 years old now. And then the work that we've been doing has been contributing to the describing and classification of ecoregions and habitat mapping within South Africa. So this just shows how these are kind of the outputs from the National Biodiversity Assessment, and all the data collected by the platform has been feeding into the National Biodiversity Ecosystem Classification and habitat mapping. At the same time, our data has been feeding into the marine spatial planning process, and has helped to inform the design and placement of South Africa's marine protected area network, and it's continuing to do that through the MPA expansion strategy. So what's next and where to from here? I didn't really go into much detail with it, but artificial intelligence and machine learning is one of the major next steps within the field of research. We collect hours and hours of imagery data and videos that typically have to be manually processed. So you have analysts watching the videos, and this is extremely time consuming, and it's a massive bottleneck in terms of getting our data from the time that we collect it to being able to provide it to support management decisions. And machine learning and computer vision is set to revolutionize that. We have a number of projects ongoing with collaborators from various institutes and universities within South Africa working on this problem, but globally there's a lot of work happening on it as well. And then future, there are alternative technologies that we could also start looking into in terms of improving the autonomy of data collection. And the one is the autonomous underwater vehicles. These are essentially, you can program them, deploy them and program them, and they'll go off and do surveys while you or the ship can continue doing other work. So they greatly enhance the capability of doing research and covering large areas. And looking into these technologies will be one of our future objectives. Yeah, and obviously this work is all quite expensive. It's taken a lot of resources from South Africa to start developing it and implementing it. And we get a lot of support from the African Coelacanth Ecosystem Program, the South African Polar Research Infrastructure, and the Shallow Marine and Coastal Research Infrastructure, which are both programs of the Department of Science and Innovation. And then a lot of support from the National Research Foundation, both SciAb and then SciON, which is the South African Environmental Observation Network. Thanks. So I think next up, I'm just going to introduce Gwyneth, so you can talk to us about the genomics platform.
Hi, can everyone see my screen all right?
Yeah, all good, Gwyneth.
Fabulous. Okay, so I'm going to talk about the Aquatic Genomics Research Platform. And we deal with, sorry, my office phone is going off and I took it off the hook and it's still going off. Okay, so what do we do in the Aquatic Genomics Research Platform? We do everything from DNA extraction all the way through to next generation sequencing, and everything in between. So it's a wide-ranging platform, and we've got researchers from all over the country because it is an open access platform, and we also do a lot of training and upskilling of people in this field of research. Now, DNA is my business, and DNA I get very, very excited about. And so I thought in order to bring you guys all along on this journey of excitement with me, I'd share some cool facts about DNA, which just brings it into perspective how awesome we all have DNA in us and how awesome it all is. So if you took the DNA in you, each of us, and we counted the number of nucleotides or letters that make up the human genome. That's 3.2 billion nucleotides in your genome. And if you were to type out all of those letters, and if you were typing 60 words per minute, 8 hours a day, it would take 50 years for you to type out the sequence that's in each of your cells. And even more awesome is that humans are not even the best in terms of number of nucleotides they've got. For example, the marbled lungfish has 133 billion nucleotides in its genome. If you took one of your cells from your body and took the DNA just out of that single cell and uncoiled it, that strand of DNA would be 1.8 meters long. And if you took all of the cells in an average person's body and put them end to end, the DNA end to end, you'd end up with a single strand of DNA that is over 1 billion kilometers long. So that's like taking 150,000 round trips to the moon and back. And this is DNA that we each carry in ourselves every single day. And the amount of information that's encoded on that DNA is phenomenal, and we're only just scratching the surface. And so this is the field that I play in, and it's a very exciting and fast-moving field. So how do you go about analyzing data? Now, the first thing you have to do is actually extract the DNA from the cells themselves and then prepare them in a format that will allow you to determine those nucleotide sequences. And there's a variety of different methods that you can use, and you can do pretty much all of them here at SciAb. Once you've got your DNA prepped for the sequencing analysis, there are different platforms available to sequence them. And we've got 3 of these here at SciAb. We've got Sanger sequencing on the Genetic Analyzer 3500. And that's more for single species or pure cultures. We can do 192 samples on a single sequencing run. And the number of nucleotides that are sequenced per sample is between 1 and 800 base pairs. Then the 2 next generation sequencing platforms we've got is the Illumina MiSeq, as well as the Nanopore MinION and P2 Solo. Now, the Illumina MiSeq is absolutely fabulous for microbiome studies, metabarcoding, microsatellites, SNPs, etc. And what it does is it can sequence up to 20 million different pieces of DNA in a single sequencing run. And the fragments of DNA range between 100 and 400 base pairs. So you can see the scale of difference in terms of these 2 sequencing platforms. The nanopore, depending on which chemistry you use, you can have between 8 to 30 million different pieces of DNA being sequenced. But the bonus about nanopore is there's no size limit to the fragment of DNA that can be sequenced. And nanopore is really, really good for whole genome sequencing, which we'll touch on in a little bit. So some of the current projects that are running at SciAp, they are hugely wide-ranging, and I definitely can't go into all of them. It'll take me 2 days to explain them all. But some of the more common ones are phylogenetics, population genetics, metagenomics, metabarcoding, microbiome research, transcriptomics, marine natural products, whole genome sequencing, environmental DNA, etc. So there's tons of them that we work with. If you think about it, DNA is in pretty much every organism in every single environment. So depending on your research question, DNA will be involved in that. I'm going to just go through 2 examples for the purposes of this talk, and this will be whole genome sequencing and environmental DNA. So whole genome sequencing, there are just over 2,000 different fish species in South African waters, of which 75 are endemic to South Africa. And SciAp, in conjunction with Diplomics and 1KSA, have embarked on a very ambitious project to sequence the whole genomes of as many of these species as possible. So this started just over a year ago, and so far we've got 18 fish species and 1 invertebrate species, and these are listed over here, that we have sequenced to completion or almost to completion. And what's really cool is a lot of these are actually on the IUCN Red List, either as vulnerable, endangered, or critically endangered. And the reason why we're doing this whole genome sequencing is that you can then use that information to do additional studies like population genomics, and distribution profiles and SNP analyses and all sorts of different things with that basic ground foundation information. Amongst the species that we've done whole genome sequencing includes the galjoen, which is actually our national fish. Just like the protea is our national flower, or the springbok is our national animal, the galjoen is our national fish. Environmental DNA is a completely different DNA-based analyses, and it is reliant on the fact that all organisms shed DNA into their environment all the time. So every time you walk around a room, you're actually shedding skin cells and sweat cells, etc., and DNA into your surrounding surrounding environment. And the same goes for a marine environment. So all of the fish, invertebrates, etc., in that environment are constantly shedding their DNA into the water column. So instead of either catching the fish or trying to monitor the fish in the actual whole organism, eDNA, what it does is you take a water sample, you concentrate it down by filtering it, and then you extract whatever DNA has been shed into that water column and sequence that DNA. So in a single water sample, you will have DNA from whatever fish or whatever organisms were present in the surrounding area. So as with everything, first step, extract your DNA, and then depending on which research question you want to ask, whether you want to look at all the fish population or a single species, or whether you want to look at algae or phytoplankton or microbes, you can look at anything that occurs in that water, in that environment, because their DNA will be in the water sample. All you've got to do is choose which primer set you're going to use to target that particular group of organisms. So just as an example for a project that we've been doing recently in the last year, we're looking at the fish population in Isimangaliso, which is a marine protected area, which Ant mentioned in his talk. And with a water sample, these are all the different species that were identified. And here's some pictures of them as well. eDNA works really, really well in conjunction with BRAV work, video analysis. Sometimes with BRAV works, it's very difficult to identify a fish morphologically, and this is where eDNA can complement BRAV's work really nicely, because each organism has its own barcode. It has its own unique DNA sequence, which allows us to differentiate between different species. The other advantage is that it can detect rare species. There are some drawbacks, and if you have any questions, I'll happily go through those as well. So yes, it works really, really nicely paired. It can't replace, but it can definitely pair up and work well in conjunction with other methods. So I've spoken at looking at the fish population as a whole, but the alternative is if you want to look at a single target species. In this case, you would design a primer that's specific only to that species to detect the DNA just of that particular species. And an example would be your coelacanths. Coelacanths were in the fossil records, and they thought they went extinct 65 million years ago. But in 1938, the first living organism of coelacanth was found off the South African coast, so it's classified as a living fossil. And there are only 2 species of this extant species, i.e. living species of this genus, and that's Latimeria chelumni of South African coast and up the coast of Africa, and then the coelacanth that's found in Indonesia. And very important is they're critically endangered, so there's very, very few individuals in the wild, or anywhere for that matter. And the difficulty with doing research on this particular species is they found not only that there's few in number, but they're found in very, very deep waters, and they hide in submarine caves. So finding them is challenging. So the idea was to see if eDNA could pick them up more easily. So the study looked at Jessa Canyon, where we knew there were coelacanths present, as well as the site upstream and downstream of that location to see if we could pick up any coelacanth DNA in the water column. So we collected water and did sequence analysis, and happy days, we found coelacanth DNA in the water column. So this proves that this methodology does work. And if you want to read up more about it, this paper is in press and will be out very, very shortly. Now, the applications of doing DNA work on any marine environment are hugely wide-ranging. It can be used for species distribution profiles. It can help in monitoring and managing marine protected areas. It can be used to inform aquaculture questions and improve aquaculture, marine ecosystem assessments, conservation of endangered species, tracking genetic diversity and population structures. It can be used in marine natural products research or bioprospecting. With the microbiome work, you can look at water system health, the presence of pathogens, pollutions, etc. So quite literally, the possibilities are endless. There's all sorts of things you can do. If it's got DNA in it, you can research that question. And there is a lot of participating organizations with this type of research, and these ones are just the ones that are involved in those 2 projects that I highlighted recently. Thank you. And with that, I'm going to hand over to Taryn.
Okay, thank you very much, Gwyneth. Okay. Sorry, just waiting for the screen to share. There we go. Clear?
All good.
Okay, fantastic. Hi, everyone. Thanks very much for taking the time. Yeah, Ant and Gwyneth covered their platforms really, really beautifully. And following on from that, I'm Taryn Murray. I'm the instrument scientist that manages the Acoustic Tracking Array platform. And we're essentially interested in finding out where animals move and why they move. So when you think of studying movements of animals, your mind might automatically jump to things like radio-collared wild dogs, or, you put tags on birds and you see these beautiful maps of these animals moving across various habitats, across continents. I mean, you get these birds that migrate between Europe and South Africa. So you get a really good understanding of what land animals are doing. But things are always a little bit trickier when it comes to water. And one of the tools that has allowed researchers to really broaden their knowledge on movements of aquatic animals is a method called acoustic telemetry. So acoustic telemetry is one of several different types of methods that you can use to track aquatic animals. But in a nutshell, it essentially consists of this thing called an acoustic receiver. So that's how big it is, and that's what it looks like. And you get a whole bunch of things called tags or acoustic transmitters. So they range in size from really, really small ones, probably about 0.5 centimeter, all the way up to about 5 centimeters. As you can see in the top right image there, I've used the standard size scale of a standard highlighter just to kind of indicate the range of the different tag sizes that you get. But very briefly, you take your equipment, you take your receivers, you can put it in estuaries, or you can put it in freshwater, or you can put it in the sea, in the inshore area, which is where we mostly study the animals. You then go and do the really fun bit, which is going and fishing, and you catch your animals, whichever species you are wanting to study the movements of, and you actually do a little operation on them and insert that tag into their body. So this tag then remains within that fish and sends out a sound signal. And once it's in the detection range of an acoustic receiver, that receiver hears that transmission and it goes, oh, okay, I know that it's this tag number of that fish, and it records the date and the time. So you really get an idea of where a specific animal is at any given time. And lucky for us and for the ATAP, we find ourselves in this real golden age of animal tracking. Acoustic telemetry is now considered one of the most popular methods across the globe to study the movements of aquatic animals. So we're really in a good position to kind of drive this research forward. So as I mentioned, you go out into the field and you tag your animals, and these can range from different types of fish to stingrays, to skates, to sharks. And currently the public aquaria at Two Oceans Aquarium, Bay World, the East London Museum, and Ashaka Marine World, they are beginning to acoustically tag turtles, which is really exciting stuff. In terms of the deployment of receivers, as I mentioned, you can basically put your receivers wherever you would like to study your animals. So this can be in estuaries. As you can see, we have lots of fun swimming around in very turbid systems. But in the estuaries, you've basically got a big cement block. You put your receiver in, you line it up with a very distinct landmark, and you leave it in the water at least a meter at spring low tide, so that you know that boats will go over the equipment with no problem. And in the marine environment, you rig up very heavy moorings and you drop them down drop them down into the sea. And a lot of our receivers have something called an acoustic release at the bottom of them, and you can pretty much try and get these receivers back to the surface remotely, so you don't actually even need to dive, which is fantastic when you're placing these pieces of equipment in slightly deeper waters than is what is with that falls without of diver safety depths. And you can also place these acoustic releases, the ones that you don't have to go dive on, in maybe more dangerous areas or areas of concern. So it's really, the changes in technology have really allowed us to expand where we're able to deploy these receivers. So by placing the receivers in these really strategic locations, you can really start answering questions. So you've got receivers in estuaries, you've got receivers in sea, and you've got fish that you've tagged, or whatever animals, moving around, sending out their sound signals. But you can, and as I mentioned, you record, all the receivers record the ID, the date, and the time. You can start looking at things like, when are fish in estuaries? Where are they moving? Are they moving up and down? Are they moving between the estuary? Are they moving out to sea? And when you've got these receivers all along a coastline, you can start getting better understanding of how an animal is using the coastline when it is in certain areas and how fast it might be traveling, all those kinds of things. So really piecing all these little movement pieces together give you a greater understanding of the movement puzzle as a whole. So this is just a little animation showing you the kinds of information that you can get from piecing all this information together. This was a study that was done on large adult dusky cob that were tagged down in the Breda Estuary in the Western Cape. The black dots indicate fish that were tagged in the De Hoop MPA, which is just to the left there, indicated by the red boundary, and the colored dots indicate ones that were tagged in the Breda Estuary. And some of them obviously are not moving on land. That's just the shortfall of the program. But what's very evident, or should be very evident, is that the black dots are hardly moving. So those MPA fish are staying very much within the MPA, where you've got your fish that were tagged in the estuary moving far further and far more frequently than those in the MPA. So you can start getting a better understanding of how what you would assume to be the same population might actually be doing very different movements. So lucky for us, we have the Acoustic Tracking Array platform, and this is what the network currently looks like. It comprises about 350 acoustic receivers, and we cover about 2,200 kilometers of the South African coastline, from St. Helena Bay in the Western Cape all the way up to Santa Maria in southern Mozambique. We've got receivers that are deployed in estuaries, as well as many, many more that are deployed in the inshore coastal environments. We currently monitor the movements of 39 species, including, 42 species, 42 species. This includes 13 species of fish, 17 sharks, 8 stingrays, 1 skate species, and our 3 turtles. Together, the Acoustic Tracking Array platform was officially formalized in October 2011. So we are 13 years into the making, which is an incredible position to find ourselves in. And together, during that period of time, the receivers have recorded over 25 million individual detections. So this represents a really significant amount of data. I really need to point out that this network is very collaborative in nature. Without these partners, who they themselves contribute time and equipment to making the whole platform, without their assistance and their help, this would not be as a successful platform as what it is. So I really just want to take time to acknowledge all our deployment collaborators and all our ATAP users and partners. Okay, so what can all this data tell us about the movement of animals? We can start understanding things like habitat and resource use. So where are animals spending a lot of time? How are they actually using environments? For example, if you consider estuaries, are these animals moving up and down the estuary? Are they moving with the tide? Are they staying in one place? Do they leave the estuary at certain times of day? Do they leave the estuary or enter the estuary at certain moon phases? So you can really start teasing these kinds of things out. You can start getting an idea of residency and site fidelity. So residency, are they staying in one place all the time? And by site fidelity, if they leave, do they come back to the same place? We can look at estuary marine connectivities. How often are animals moving into estuaries or into the marine environment? And how often are those connections being made? We can start understanding longshore movements. So are they moving across hundreds to thousands of kilometers along the coastline, or they may be only moving about 5 kilometers? What about seasonal migrations? You can start picking up when animals are moving up and down the coastline, and highlighting, or it shows you the importance of certain areas at certain times of the year. You can start assessing things like population dynamics. And if you know the sex of an individual, unfortunately, in many of our fish, we can't sex externally, but for many of the sharks, you are able to, the sharks and the rays and skates, you can externally sex them. So then you can start getting a better understanding of whether females do one kind of movement and males do something else. So this is really exciting stuff that all adds to the knowledge that we know and about how animals move in certain ways. So why is all this information even necessary? I'm sure there are some people that have joined us that are avid fishermen, and at the end of the day, fish and the aquatic resources represent important food resources for people. And there are many, many individuals that rely on these resources for income and for their livelihoods. So a lot of the species that we study are of either commercial, recreational, or small scale importance. So it's important to understand the movement of these individuals to try and better manage them, but to work together with the representative parties just to try and come to some kind of common ground, because at the end of the day, what we're trying to work towards is protecting individuals so that there are more individuals, which leads to pretty much fish for everyone. So that's kind of the direction we'll be going with. So as I mentioned, the data can be used specifically for things like fisheries planning and management, and very, very much spatial planning, because at the end of the day, what you're getting, what a metric that you're getting from using acoustic telemetry is, are they there or aren't they there? So this is really, really important for spatial planning. You can start thinking of things like if there are multiple species tagged in a very small area, how important is that area for all those individuals? So this is really, really important when it comes to spatial planning. And a quick visual to try and explain this a little bit better is, if you've got a species that pretty much sticks in one place all year round, you could slap a marine protected area or estuarine protected area around that population, and you know that 99% of the time they're going to be in pretty good condition and they won't be targeted, they're going to be protected. But a lot of the time fish don't do this and they tend to move. You might have some individuals that feed in one place and head somewhere else in winter to do an activity like spawning, where they go and they lay their eggs. So you've got this movement up and down the coastline. So having an MPA in the middle is doing nothing. You're not protecting those animals in any way other than when they're swimming through that place. So then you might have to change tact and think about adding, smaller MPAs in very important areas. So the importance of this technology to the acoustic tracking or acoustic telemetry field is the changes in technology have been absolutely incredible for this field of study. First of all, the average lifespan of these tags can range from 2 months to 10 years. So you can start tracking individual animals for up to 10 years of their life, which is unbelievable. And believe it or not, there are actually some individuals that have been tagged, which include learfish, which is an important recreational fishery species, and ragged-toothed sharks that have up to 10 years of data for some individuals, which is just absolutely incredible. We're now able to study smaller individuals because with the the diminuization, I think if that word is correct, so basically tags are getting smaller and smaller, which are allowing us to tag smaller and smaller animals. So we can start getting a better understanding of movements of really small juveniles. Unfortunately, you don't quite get that move as they grow because the tags are so small, but still important information. You start getting fancy things like predation tags. So these essentially look exactly the same as these ones, but they've got a little sensor on them. So if a predator eats a smaller individual, that little sensor actually has like a little seal on it. The stomach acid in a predator will melt that seal, and it will cause the tag to change its ID code, which means it's then sending out a new ID. So when a receiver picks up that signal, it records that new ID, and you know that that fish has been eaten by something. So there's some really incredible developments happening, and yeah, so much more all the time. And with that, thank you very, very much for listening. A big shout out to the ATAP community, all our deployment collaborators as ever. very much the National Research Foundation and our Canadian-based partners, the Ocean Tracking Network, for our very much the backbone of our infrastructure platform. And without the assistance of the African Coelacanth Ecosystem Program and Save Our Seas Foundation, we would not be able to cover the running expenses of the platform. So very much thank you to both those funders. If you ever need to contact us, please follow us on social media. I can't promise we're overly active, but we try our best to keep some content updated. And please feel free to reach out if you have any questions. Great, thank you very much.
Thanks to the speakers. Those were very interesting talks. It's amazing to see how technology allowing for such detailed information to be gathered and in places that we haven't been able to gather information before. Yeah, quite amazing. If you've got any questions, please, you can use the question and answer function, or you can post them into the chat or raise your hand and we can unmute you. And you can ask any of the speakers a question. I've seen there's a couple in the chat already. We've got Anila asking, Gwyneth, have you worked on shrimp identification with the eDNA before?
Specifically, no, but it's very easy to use our current pipelines and just make sure that all you got to do is change the primer that you're targeting. So at the moment, no, but we're hoping to move into that area soon.
Karen, Donnie's asked what the range is between tag and receiver. What's the listening range?
Great, thanks, Ryan. Hi, Donnie. I replied to you in the text, but in case anybody's interested, the detection range of a receiver. So a receiver like this can only listen a certain distance, and it really depends on the environment in which the receiver is deployed. So in an estuary or even a freshwater system, you might have a detection range of 50 meters to about 600 meters. So it depends on how many rocks are in the water, how many water weeds are there. At sea, if you go into maybe like 2 k's from the shore, or not even like a kilometer from the shore, your detection range is significantly bigger, which is about a kilometer, roughly 750 meters to a kilometer, but it can be even higher than that.
Thanks, Taryn. Shannon has asked, is there progress towards developing more MPAs in South Africa? I think Ant will probably be best to answer this.
Yeah, thanks, Ryan. Thanks, Shannon. Yeah, so the Department of Fisheries and Oceans and Coasts, together with SAMBI, are undertaking a big spatial prioritization and spatial planning initiative to identify new MPAs and expand the current MPA network. I think the target for this next, I might be wrong, but I think the target for the next phase of MPA expansion is 20%. And for the most part, these are looking at more offshore environments, so deeper water environments. Yeah, so there's a big process happening within South Africa at the moment to strengthen the MPA networks. Thanks.
Thanks, Ant. I see we've got a hand up here, Sinotando. We unmute Sinotando and she can ask a question. How do we unmute Sinotando?
They can unmute themselves.
There you go.
I think Sinotando should go ahead and speak. The mic is unmuted.
Cosmo, would you like to ask your question? Okay, we'll go on to more questions from the chats. Ralph's asked Gwyneth, what's the range for eDNA sampling, and how strong is the influence? Is it influenced by current? For example, with the coelacanth DNA, how likely is it the detection in the southern site is due to the DNA being carried down current?
That's a very, very excellent question and a very valid question. The range for the eDNA sampling, it really depends on a lot of different factors because DNA doesn't last forever. It gets degraded and microbes actually eat it. It depends on the environment it's in. So things like turbidity, microbial load, UV, etc., the temperature, all of these will affect how rapidly that eDNA will break down. eDNA doesn't, on average, and this obviously differs between different environments, doesn't last more than a day. So it doesn't last forever. And yes, it can be washed down by the current, so that is something to keep in mind, particularly in the coelacanth example. But the reason why we don't think it's a current issue is because there were a lot of other sites that I didn't mention where we weren't in between those two, where we didn't pick up any coelacanth DNA at all, which what makes us think that that second site where we haven't found coelacanths visually before is a legitimate coelacanth detection itself. Also keep in mind that the current along that coastline is very fast, but dilution and breakdown of the DNA will account for most of that DNA being destroyed before it gets there. Does that answer your question, Ralph?
Thanks, Gwyneth. Got another one for Ant here. What are the metrics used for assessing the effectiveness of MPAs, Ant?
Yeah, thanks. So there are a variety that you can use. It's largely dependent on what the purpose of the MPA is. So MPAs get proclaimed for different reasons. It might be protecting ecosystems or biodiversity or providing fisheries support. And so the metrics will vary based on what the purpose is. And then there's multiple dimensions to MPA effectiveness. So it can be from the recovery of biodiversity or the recovery or the maintenance and recovery of biodiversity to the recovery of fish stocks, but it can also reflect the level of compliance, the interactions with surrounding communities and society, and the acceptance of MPAs by society, by the public. From in the space that we deal with, a lot of the work is with fisheries targets, and having an understanding of the populations inside the MPAs relative to outside the MPAs is a really useful tool. We've been focusing on looking at the spawner biomass, or the number of potential spawners within the MPA, as an indication of primarily the reproductive potential and the potential contribution of these MPAs to areas outside. So the biomass of mature fish, for example, or mature fisheries targets. But then there's an array of diversity metrics that can reflect the status of biodiversity within the MPAs, and you can track that through time. And you can apply similar types of thinking to other things related to the habitats and the ecosystems, so the health of the ecosystems in general. So yeah, there is quite good literature out there on the different types and the options at the moment. Yeah, ultimately it will depend on what the purpose of the MPA is. Thanks.
Thanks, Ant. Doesn't look like we've got any more questions in the chat and no hands. Karen, I've got a question for you. We hear a lot about sound pollution, and obviously your equipment uses sound. Does this add to the problem? Are other fish or animals able to detect or hear the tags? And might this impact your tagged animals?
Okay, cool. Thanks, Ryan. Yeah, we get asked this question quite frequently. So the tags that we operate with are sending out signals at 69 kilohertz. And from the research that we've done and the literature that we've read, this doesn't necessarily impact on the frequency is too low for some of the animals to hear. So while the sound is out there, it's not really impacting on other animals. There have been some studies done on gray seals, interestingly, which seem to be able to learn behavior to try and track animals. But this was also within like a small pool environment. So I think they would get attuned to that signal far quicker than what you would in a normal natural environment. So from what we can tell, no, the impact is really, really like completely negligible. So it's not really contributing to any noise pollution at all. What's really cool is that some of the receivers actually record noise, so we can start getting an idea of how much anthropogenic or like people noise is in the water at any given time. And this will obviously impact how well the receiver can hear for the tag too. So you can start sort of tying together anthropogenic noise, so noises from boats and industry and whatever else, and link it to the presence of certain fish in certain habitats or environments, because there are a couple of species that we have tagged, like dusky cob and spotted grunter, that make noise themselves and communicate through noise. So it's nice to be able to kind of link the two together.
Thanks, T. Okay, there's still no more questions in the chat or Q&A, and it's almost 11 o'clock. Time we'd set aside. So yeah, just thanks to the speakers for the very informative talks. Thanks to all of you for joining and engaging with the presenters. Oh, we've got one more question here. Thank you again for Ant. How do MPA networks help in mitigating the effects of climate change?
Thank you. So there's the effect is not always direct, I guess, and they're not often set up for that. But there has been quite cool research recently, some undertaken by Rhodes University and the Safer Lab. Yeah, the Safer Lab, demonstrating that the MPAs preserve the more resilient and more fit members or phenotypes or genotypes, I think. I don't know, I'm probably not the best person to answer this question. But they basically, because they preserve those types of genotypes, the fish within the MPAs are better able to cope with the stresses that will be imparted on them by the climate change, so by increasing water temperature. So in theory, the MPAs will be another refuge of resilient fish, or fish that are more, or individuals that are more resilient to climate change. So that's the one of the main thinkings now. But at the same time, having a more complete ecosystem and a more naturally structured ecosystem, ultimately with higher biodiversity, ultimately increases the resilience as well. So that we're better able to cope with the changes that might come, or the impacts that might come from climate change in comparison to areas that are outside, that have been heavily degraded already through human activity.
Thanks, Ed. I hope that answers the question. Yeah, I think we'll wrap up there. Yeah, thanks to everyone for joining, and for to the presenters for their time and putting together such informative and interesting talks. Thanks.
Thanks, Ryan. Thanks, everyone.
Thanks, Ryan. Thanks, everyone, for listening.